I'm very pleased to introduce Dr. Peter Forster, who is geneticist at University of Cambridge, as well as Director of Research at Institute for Forensic Genetics in Münster. So as we have now a lot of colleagues from our brothers and sisters from different Rotary clubs from around the world connecting, let's just start with a small introduction of Vilnius International Rotary Club. So the president, Laimonas Markauskas.
Thank you, Vilja, and thank you, Peter, for joining us today. So just a couple of minutes. So welcome everyone to our Vilnius International Rotary Club meeting with very, very interesting guest and interesting presentation. Shortly, for all our guests, we are one of two international Rotary clubs in Lithuania. International means that we have English language as our common language when we have all the meetings and everything. And as all Rotary clubs, we have some permanent yearly projects which we do each year. So in addition to many ad hoc initiatives which we do, but we have two main projects, and these are Children's Summer Camp and Let's Help Stars to the project Let's Help Stars to Shine. So Children's Summer Camp, it is a camp, 1 week camp for children who are coming from kind of need, because they're coming from social risk families and the like. So they spent 1 week in the camp, have quite nice trainings, entertainment, and many good other things. So this year we are still planning to have a camp, although we have no idea how the situation goes, but the work is on. Last summer it could be possible, although we had to skip it, and I hope that this summer it will be possible anyway. For this, we are collecting our budget and kids as well, because not only money, but kids who would come to the campus sometimes an issue. So I encourage all the guests to support the camp and help us, if not with selecting a couple of kids, but with helping with some financial support. If you are interested, we can send you more information about it. And another project, we call it Let's Help Stars to Shine. This is the project to help young musical talents. So we cooperate with Rostropovich Foundation, who helps us to select 2 kids for whom we provide a scholarship, either for some studies in abroad schools, or for example, to buy instruments which are usually quite expensive. So this is what we mainly do. These pandemic times are a bit different to us, but we are trying to continue as much as we can, maybe in different formats. So that's about our club.
So thank you, President. So as you know, today we have a very hot topic, tracing the origin of the coronavirus. So let's dive deep into the waters of scientific world. So Dr. Peter Forster, the audience is yours.
Okay, thank you. Right, so I'm going to talk about the origins of the coronavirus genomes, in the sense of when they first successfully spread amongst humans. And in order to find out where the origins lie, I analyze existing coronavirus genomes using a method which we developed, which we call phylogenetic network methods. And therefore my title is Phylogenetic Network Analysis of SARS-CoV-2 genomes. So let's take a look now what this means. Oh, just a little bit about myself. As Vilja said, I'm a geneticist at the University of Cambridge. At the same time, and there I use my genetics in archaeological applications, ancient DNA, for example. and I'm also a director of research in forensic genetics, which has applications. Right. So let's start 30 years ago. When I was a student, I was interested not only in my chemistry studies, but also in human prehistory. I wanted to trace where humans come from and how they spread across the world. And at the time, you possibly heard, geneticists, a New Zealand geneticist working in California, Alan Wilson, he had discovered how to use parts of the human genome to reconstruct a family tree of DNA. And with this family tree of DNA, you can see which humans moved at which time to which continent in prehistory and colonized our planet. Now, he presented the method, the biological basis how to do this, but in 1990, this professor in California did not have a method how to reconstruct from his data the tree which connects together all humans. And they tried various what you call tree building methods, and it didn't work very well. But I had a look at the data, and I got together with a mathematics professor in Germany, and we worked out two methods together how to reconstruct reliable evolutionary trees from DNA data. And thus we were able, with this phylogenetic network method, which we invented, we were able to discover the movement in prehistory of humans coming out of Africa 60,000 years ago, moving into the Middle East, and then colonizing Europe 40,000 years ago, and the Indian subcontinent, Australia, and then finally America 20,000 years ago. So all this we were able to do because we had developed this network method. Our last publication in this was, I think, in 2007, where we described the first settlement of Australia 50,000 years ago. So all this is based on DNA analysis of modern DNA. With modern DNA, you can use our method, phylogenetic networks, to reconstruct what previous DNA, which is now no longer existing, what that DNA looked like. Now, with this background, I heard or I saw in the news in January last year that there was a virus in Wuhan in China, which was spreading. And I knew immediately that the methods we used for human prehistory, for tracing how humans spread, can immediately be used for tracing virus genomes to see how they are spreading. Now, what convinced me that I need to publish quickly was my experience as a student in virology in Germany. I was in a top institute there, excellent virologists, the best in Germany. But what I quickly noticed in the 1990s, these virologists did not have training in data analysis, in evolutionary data analysis, because DNA sequencing was only invented a few years earlier. And in their training, there was no DNA, there was no phylogenetic methods. We had invented all this new. And I thought, if we now have a virus coming out of China again in 2019 or 2020, and the virologists do not have good methods at their disposal, then the virus could spread everywhere and we will be in trouble. And I had already noticed in January the mortality figures were unusually high for this new virus. So what do? You have the method, you have the knowledge. What you need normally is to apply for research funding and to get a research team together with this funding and then analyze the data and publish. But I knew we don't have time for this. So instead of the research team, I recruited my own family. So my wife is a medical doctor and she worked with me for her PhD on DNA analysis. So she's an expert. My brother is a cancer genomics expert. He then also has the knowledge how to analyze viral DNA or viral RNA, as in this case. And then my professor supported me throughout, Professor Renfrew, who has the knowledge from anthropology of what I was trying to do. So the four of us worked day and night. And very quickly, this is what happened. So the first virus in Wuhan, the first genome was sequenced on Christmas Eve, 2019. Then data were provided mainly in the beginning by Chinese geneticists who provided data on a database. And on the 3rd of March, we took what was available on that database, about 230 virus genomes, and we sorted out the best of them. That was about 160, which were good quality. And we analyzed in 2 weeks the data using our phylogenetic network method. So by March the 17th, we were able to submit our manuscript to a journal called Proceedings of the National Academy of Sciences and of the USA. Then in these publication proceedings, you have what is called a peer review system. So your manuscript goes to experts, and these experts then have to decide whether what you are publishing is reliable or not. And the experts then criticize, so you have to change your manuscript, and then finally you publish. And because it was coronavirus, the journal processed this very quickly. And on April the 8th, we published, and we were the first in the world outside China to publish the evolution of the coronavirus. And this paper is now very highly cited, over 600 times, and every day more and more researchers are citing it in their own publications. So that's just the history of it. Now let's see more in detail what we did. So we applied our phylogenetic network analysis to 160 of the first SARS-CoV genome sequences, which were taken from patients between Christmas 2019 and the 3rd of March 2020. And if you look at this diagram, it shows you this circle is a particular virus genome type. It's a big circle because, let's say, 6 or 7 different people have it. And in this small circle, only one person has it. So the size of the circle show you how many patients have this particular virus type. The color shows you the geography. Yellow means it is a Chinese patient. Orange means it is a different East Asian patient, for example, Japan or Taiwan or Hong Kong. The blue colors are Western populations. So that is Australia, North America, Europe. The lines connecting the circles, these are the positions at which the virus genome has mutated. So you see this type down here, yes, differs from that type with the A by one position here, one link. And this could be, for example, position 8,292. So the Chinese down here has, for example, at position 8,292 an A, and the people up here might have a T at that position on their DNA. Yeah. I say DNA, actually, technically it's an RNA virus, of course. So this is how the network works. You have these circles representing viral genomes. You have the colors representing where the people are from, where the patients are from. And then you have these links which show what is mutating and at which position precisely. And what we saw immediately is summarized here. We find 2 clusters, A, we call this, and B up here, and then a smaller cluster, C. And all these types are common in China, as you can see, because all of them have some yellow proportions in them. But also you can see at an early stage, you see the blue types coming up, and that means that the spread happened quite fast also to the Western world. So the first thing you do when you have such a network, at least when you study human evolution, you want to find out where is the original ancestral type. And you do that by applying what is called an outgroup. That means a virus genome type which is certainly branched off before the virus differentiated into virus subtypes. And what can you take as such an outgroup to find out where the root is? Well, you can take the bat. So down here, I'm sorry, I just have to move my slide here. Down here, where the long blue arrow is pointing, is the bat coronavirus. This is 96% similar to the human coronavirus. And it clearly shows that the oldest virus type is the type A. And that is a surprise because the type B is the common type in Wuhan. Type A is not common in Wuhan. And at that time, I was surprised because the media story was that the virus had spread from the fish market in Wuhan. And that was the origin, possibly because some animal in the market carried the original virus, and that then spread to humans. But if that story is correct, why is the majority of virus type in Wuhan in this early phase, why is it B type and not A type? We find the A type, in fact, is more common in other parts of China at this early stage, for example, in Yunnan, and for example, in southern China, in Guangdong, which is close to Hong Kong. And what is interesting with this observation is that the city of Wuhan does not really have a bat population where the virus is thought to come from, but Yunnan has those bats, and Guangdong also has bats. And Guangdong in particular has a higher frequency of the original A type, and it is also where two other coronavirus outbreaks happened. The first SARS outbreak in 2002, and then a coronavirus outbreak among pigs, also from Guangdong in southern China. And this would mean that this current coronavirus outbreak is the third one from this area. So that is my speculation. And I think it is well worth looking at the origins again more closely to see what is causing these continuous outbreaks. It does not seem to be the fish market, that for me is for sure. Right. I'll now show you the next slide. So here you have exactly the same network, but this time the colors are not geography, but they are time. And I wanted, so for example, the earliest sample is from Christmas 2019, and it's sampled here in week 0, as you can see on the left, and that is in brown, and this first sample is up here. Then in week 1, you have this gray color, so you have gray here and here and here. And then you have white for week 2, yellow for week 3, and so on. And if you look closely, you will see that you cannot see anything because the time of the sampling, the week of the sampling, shows you no clear pattern. It is a big mess. And this is an important lesson because what I used to find out the original sequence type was the BAT outgroup, to find out that A is the original type. But that at the time was not the usual way how to find out the origin of the pandemic. Some virologists were arguing, well, the oldest patient in my sample must have the oldest virus type, and therefore that patient will represent the root of the pandemic. And as you can see, that is totally wrong, because the oldest patient, 0 in brown up here, is very far away from the root as indicated by the bat. And I was able, I think, through our publication to convince the majority of virologists that you need to use a proper statistical method to find the root and not simply take the oldest patient. That is misleading and it's statistically not reliable. There is still a large minority of virologists who have not yet taken this message on board, but I'm pleased to say the majority, certainly of European and Chinese virologists, they have followed this advice. Right, now this is the same network again in this slide. And this time, I am discussing the mutations in detail. So up to now, I've discussed the geography, I've discussed the time, and now come the mutations as such. Now, you have to know that this network has what we call these squares or reticulations, these diamonds here and here and here. And this is what distinguishes a network from a tree. A tree will give you a single solution. If you take one data set and say, make a tree to your computer, it will make one single tree. This network, however, is a combination of different tree solutions. And this is the strength of this network method. You see here, at one glance, because of these rectangles contain several possible trees. And in total, you see here in this simple diagram, 288 mathematically optical trees, all at one glance. Imagine if I show you 288 trees on this slide, I would show you many, many, many slides, and you would be totally confused, and you would not see the wood for trees. And this is the strength of the networks. You see all the tree solutions at one glance, and that is how we were able to unravel human prehistory, colonizing our planet 60,000 years ago. And that is why we are now in a position to understand what the virus is doing. And for one detail what the virus is doing, let me show you this rectangle or this ladder of rectangles is created by one particular position, which is shown where the blue arrows are. So this parallel mutation is the same as that one, is the same as that one, is the same as this one, and it is position 11,083. This position on the virus genome is mutating again and again. We see that also in bigger data sets, it's the most unstable position of the virus. And that is useful information because it tells us if you are developing a vaccine, please avoid this unstable position because the virus is very likely to mutate there and then perhaps your vaccine is no longer effective. And so this kind of analysis can actually distinguish how to optimize, for example, treatment, or how to understand why is the virus mutating over there and not at this position. Now I come to the main object of my talk. The original reason why I thought it was so urgent to make the experts familiar with the network method is to trace infections. And that will give us a chance to quarantine someone, but also to find out the contacts that that person had previously and also quarantine the source of the infections. And hopefully that would spread the virus, stop the virus from spreading across the world. So let's take the first example. This is the first Canadian case. So this Canadian is a Chinese Canadian from Ontario. And he had visited, from Canada, he had visited Wuhan and Guangdong, I presume his family, and then returned to Canada and then became ill with COVID on January the 27th. His DNA or his viral DNA was sequenced and submitted to the database. And as a registered user, I used this database and found out that that is his virus genome here. And in our network analysis, if you ask the question, where did he pick up the virus? Where was he infected? You can go back one step, one mutation, and the network says there is an empty node here. The network has constructed this position where the blue arrow is pointing, but it does not exist as a type in the database because it has not been sampled, for example. But you can see from the derived types, these two yellow nodes, where the ancestral type probably came from. So one ancestral node is Fushan, and that is in Guangdong. And the other derived node is Shenzhen, which is also in Guangdong. So both these descendants indicate that the ancestor was also in Guangdong. And that is where the patient had traveled to, Guangdong, you can see over here. So this is a nice way how to find out where someone got the infection from. And if you know that, then you know where to start your quarantine efforts. And the story does not stop there. As you can see, soon afterwards, this Canadian must have infected another Canadian. There are 2 mutations that happened between this patient and that patient viral genome. And then also 3 Californians have been infected by this type from Ontario. So you can quickly see how this virus spreads if you just look at this network analysis. So let's take a look at the next example. This is the first Brazilian case. The Brazilian case is shown in green down here. Let me move my slides over there. So the history is that the Brazilian patient had visited northern Italy, that is Lombardy, as a tourist, and then became ill on the 25th of February, a few days later. Now, analyzing him in the network, you can see there are 1, 2, 3 mutations back, and you arrive, in fact, at an Italian. Or in fact, this is a larger circle. These are 2 Italians who have the same virus genome. And then there is a derivative here, also another Italian. And that means the network confirms that the Brazilian picked up his virus from northern Italy. But now comes the next question. If you want to find out where did the Italians get their infection from, the network says it is not clear. Either you can go along here, this is 1 mutation happening, and this is a type which is found, for example, in Singapore, Hong Kong, and so forth. Or you go back this way, and in that case, the Italian picked it up. This is an Australian Chinese in Sydney. Sorry, a Chinese Australian from living in Sydney, but visiting family in China. So that is the other possibility. And this is also the strength of the network. If the data are uncertain, it will show you the 2 or 3 different possibilities. If you just provide a tree, it will show you 1 tree, and the tree will say, and this link here is uncertain, and that's all I can tell you. But the network actually shows you all these various possibilities at 1 glance. Right, now we come to the most scary slide, and that is the question, where does the globally dominant virus type come from? So the virus type we have today worldwide, where does that come from? So we all know, if you think back, that the global pandemic really started with the spread of the northern Italian viral type. So that is the type shown here with this blue arrow, the first case in Lombardy in Italy, that blue circle. Now, the network links a type before that here to Germany, and specifically Munich. This Munich infection happened because a visitor from Shanghai, a lady, had visited on January the 19th, 2020, and had a working visit with several German engineers and infected a few of them. She stepped on the plane back to Shanghai after her work visit was over, and then on the plane she noticed she was getting ill. And as soon as she landed, she went to the doctor, went to hospital, and then informed her German colleagues, I am positive for the virus, you had better take care. And then immediately the Germans tried quarantine on these colleagues, and officially the quarantine was effective, and the virus outbreak in Munich was contained. The alternative view is that the quarantine was not quite effective because you see this close link to North Italy. And what we found is that of the 16 people quarantined in Munich, one of them went to the Italian border, just a few kilometers from the Italian border. And then 3 weeks later, or less than 3 weeks later, the first Italian patient showed symptoms. So one version is that the Italian and the original source of this Italian infection has not been traced in a documented way. This is a genomic tracing, but it has not been traced by a personal interview. So they tried to search for the contacts, but they failed in Italy. So one view is that the Italian type came from China directly, an independent visit. That does not seem very likely because the Italians had a flight ban. So all flights from China were banned from the end of January onwards. And the other possibility is that the quarantine was not effective and that we have this infection across the border into Italy, because this is, after all, the skiing season. All right, now the story goes on. We then have the spread from Italy to, for example, Mexico, the first Mexican case. And we have a spread then back again from Italy back into Germany, into South Germany. In this case, this was a German visitor in Milan. And the major viral types you know about today. So what I've been talking about are all B types. This is our A type down here. This is the B type here. This then turns into the B Shanghai type, which turns into the B Lombardy type. And from this Lombardy type, you have all the major variants you know today. For example, the Kent variant in Britain, which you've heard of, which is now spreading, I'm afraid, sorry about this, in Europe and around the world. That also is derived from this first type. It is called the B117 type. B is the same B that we labeled it here. And then you have also the Brazilian type. So these types have all evolved from this first successful branch. Right, I have here an early criticism of our paper. So because I am not normally working in virology, except my first degree was in virology, but you know, my career has not so far not been in virology. Of course, you get many people who are career virologists who are very angry that someone from outside the field has stepped in. So they wanted to show that I'm not right. And they said, don't use network methods, tree methods are better. And here, for example, you can see we've taken the same data set as Peter Forster, except we've added 5 new sequences from Holland, Wales, Brazil, and Portugal. And if you add them, then you see the whole tree becomes unstable. Yes, sometimes the Italian is here, sometimes it's down here, sometimes Wales is here, sometimes Portugal is down there. You know, each time you calculate a tree, they come up in different positions, and therefore this whole tree is unstable, the data is not reliable, and their conclusion is you cannot trace infections using genomes. That does not work. So I wrote to these colleagues who are in Florida, and I said, could you please give me your 5 new genomes? I want to take a look what's going on. And they said, certainly, here they are. So they emailed me these 5 virus genomes from Holland, Wales, Brazil, and Portugal. So there were 2 Dutch, I think. And the first thing I noticed is that these virus genomes, these 5 new virus genomes, are identical. And the tree that they use is not able to display identical genomes as one single branch. It splits it up into these deep branches, and it makes it look as if they are different genomes, but they are not. They're all the same genomes. That's problem number 1. The tree makes this quite trivial mistake. It always assumes the viruses are all different from each other. And the second problem is the researchers did not look at the travel history of these 5 people. So what they are saying is this Portuguese, Brazilian, Dutch, Welsh, these are infections all over the place. There is no particular link to this Lombardy infection in Italy. But they did not look at the travel history of these individuals. So I looked it up. Fortunately, I understand some Dutch, I can read Spanish, and therefore I was able to look up in the local reports where did these 5 people go 1 or 2 weeks before becoming ill. All 5 have traveled. The 2 Dutch people were in northern Italy. The Welsh patient, the first Welsh patient here, also had been skiing in northern Italy. The Brazilian had also been to northern Italy. Only the Portuguese, he had been to Spain. But where the Spanish person got it from, I do not know. But 4 out of these 5 new sequences had all been to northern Italy. And therefore it shows that the genomes do tell us indeed where a person got the infection from. So the conclusion that these Florida researchers has is the opposite. The genomes give a very good idea of where you got your infection from. And since then, so we answered that, and since then, I believe there has been no more criticism of our method. This was about in May or June last year. And my conclusion also is, in general, for everyone, do not only use statistics, please also use your experience and common sense when you are analyzing data. Do not simply rely on your computer output, because it can be extremely misleading if you don't use common sense. So why does this genomic tracing seem to work? Now, if you think about it, the interval from one patient infecting the next, this is called a serial interval, is about 4 to 5 days for the coronavirus. And the mutation rate of virus is about 30,000 positions long, 30,000 nucleotides long, that's the virus genome. And on average, you have 1 mutation every 2 weeks, every 15 days. So after about 3 infections, 5 days, you can on average expect 1 genomic mutation. And that means there is a reasonable correlation between the mutations happening and the patients being infected one after another. And that is why it kind of works quite well as a tracing method, to use the virus genomes to find out where your infection came from. Now, finally, the last part, I wanted to know when the pandemic or the epidemic started spreading successfully amongst humans. This is not the same as saying when did the virus first transmit from an animal to humans. That can have happened long ago, because the difference between the bat and the human coronavirus is 4%. And using our mutation rate, that's on the order of 50 years back in time to the common ancestor between human and bat coronavirus. We can discuss that more, but I'll move on to the next question. So it's not the question of when the virus jumped from animal to human, but my question is, when did the virus successfully start spreading amongst humans? So with the help of my colleague in engineering, Dr. Mike Pivnenko, we took this time 1001 high quality genomes from the early phase, December to the end of March, and did a linear regression. As you can see, it's not a very tidy data, so it's quite messy. And that means you have a confidence interval, which is also quite wide, you know, this pink area. And where this straight sort of goes through the 0 line, that is where there were no mutations, and that is where the first patient was infected and started spreading the virus. And that we have dated to between September the 13th, 2019 and December the 7th. So this time interval is the 95% confidence interval. So if I put it differently, it means there is less than 5% probability that the mutation happened after December the 5th, or the 1st, sorry, the first successful spread was after December the 7th or before September the 13th. So it's 95% confidence it happened between these two dates. And that is important to know because very often as a doctor, my wife being a doctor, is the question, doctor, I had the symptoms in 2019. Did I have the virus? And these questions come up every time that you get a media report estimating when the virus started spreading. For example, Harvard University publicized a study on hospital car parks in Wuhan, and they saw that more and more cars were parking in hospitals in China in August. And that for them is an indication perhaps the epidemic started as early as August. You've heard of the early French case already happening in December. Previously, it was thought there was nothing before January in France. Then you have the sewage analysis in Italy and in Spain. Some of the sewage analysis says that the virus is detectable from December onwards. But there is one single sample in Barcelona which says, oh, it looks positive in March 2019. Did we have a coronavirus then? The answer that we always give is, according to our time estimate, there is less than 5% probability that the virus spread successfully in humans before the 13th of September 2019. I have to say, though, we have to be careful because our estimate is based on a linear assumption that the virus mutation rate has not changed over time. But I already know that that is not quite true. If we look at viruses from Christmas 2019 up to January 2020, we see the mutation rate is on average 1.5 mutations per month. But in February and March 2020, the mutation rate accelerated to 2.1 mutations per month. And this possibly is influenced by the new Lombardy virus variant, which seems to be mutating faster. And if the virus has changed its mutation rate recently, we cannot guarantee that in the past it has also changed its mutation rate, and therefore our time estimates would correspondingly be inaccurate. So this is the best estimate we can provide. I'm not aware that other people have provided any better estimate. What we need finally is to look at hospital biopsies, where you have samples from patients from September, October 2019, and see if the virus is present in those hospital biopsies. As you know, the WHO, the World Health Organization team, has visited China, Wuhan, tried to find out more about the origins. They have recommended our paper to journalists, as I know, but they have not been able to find any deeper evidence. So the question is still to be solved. Right, so that is the end of my talk. Here are some acknowledgements, and if you have any questions, do let me know. I'll now press the stop share screen so that you can see me again. And yes, any questions, just let me know.
Thank you very much, Dr. Forster. It was absolutely interesting. And yeah, I was saying deep waters of scientific world. That's for sure for us.
Yeah.
So, but please, the audience, if you have some questions, raise your hand and unmute yourself. So we have first questions. So, but if not right now, I will ask something.
Yeah.
So you published your research already a year ago, and as we know, the also World Health Organization was looking at it, and this organization was giving some regulations, was recommending something to governments. So what do you, as a scientist, how you evaluate those measures taken by governments of different states to contain the spread of coronavirus?
Oh, that's a very sorry story. So I found in this situation that it's difficult to get people to listen to you. There are two reasons. So I have written to government officials. I have been in contact a lot with Chinese state media. I have been contacted by American officials, and it seems to me people are interested, all these people are interested in political aspects of the virus, but not in actually stopping the pandemic, at least as far as scientific involvement is concerned. And it is perhaps partly a lack of knowledge. They do not know what science can do, and therefore they do not become active. It is partly embarrassment. You know, if you have a virus escaping quarantine somewhere out of China or over the Alps or something, you prefer to shut up about it and hope nobody notices. So that's one human reaction, and therefore nothing is done. And then I think there is also academic competition where you say, you know, I can get my professorship if I publish this, and now he has published that, so that's a problem now for me. And therefore, they are not going to support you. And this is something I think which possibly explains on a broader global scale why we are still sitting here in lockdown and sensible things are not being done, because there are too many different interests at all levels. That is my private experience.
Okay, thank you. So, and still we are waiting. Okay, yeah.
I have one question. This is Aurika. Sorry,
Yeah.
I don't have a video on, but thank you for great presentation. You actually answered, I think, a lot of questions and kind of, you know, brushed off all this fake news that people are exposed in today's world. And I'm amazed, you know, of the way you created the paper by involving your family and using all the resources. So I kind of can relate my story to your story, but I have a few questions actually.
Go ahead.
So one of them would be when you were talking about the tracing the virus in Italy, I was hearing that there are some like illegal human trafficking and traveling, especially in the northern Italy, because there are some underground factories where a lot of Chinese workers would come and work. And that's like one of the also guessings why this area got such a high spike in such a, you know, short time. So I don't know if you heard about it, but just wanted to ask you if you...
Yes, I did hear about that. That was also my first suspicion. I saw it early on in January, I think in a German newspaper. They say there are many Chinese immigrant workers in northern Italy, and they often visit their family, going back and they are for low wages. They are doing, I think, traditional Italian industries. I forget exactly what it was, but some very traditional Italian industries are now being done more cheaply by immigrant Chinese labor. And that seemed an obvious link to China. Now, that may be so, but I have a database that is available for everybody, a database of Chinese viral genomes. This particular type, which you find in Lombardy in the first Italian patients in Codogno, 38-year-old, very, very seriously ill Italian. He was on ICU for many weeks. This virus type is found less than 3% in China.
Wow.
So you have to ask yourself the probability that you take the virus type once from China to Munich and once again from China to Italy, and both times you take the same virus. How probable is that statistically? And then you have to compare it with the other scenario. You have this Munich infected person coming to the Italian border. How many people can she cough and sneeze on? You know, the replication factor is about 3. And how likely is that to happen? And you have to balance these two probabilities. And as far as I know, nobody has done that yet. But qualitatively, just to go in there twice and come up with the same type is not too likely, I think.
Yeah. Oh, good. Thank you.
Yeah.
Another question was, you mentioned at the beginning that there is, important information for the pharmaceutical companies when we're developing the vaccine. So did they listen to your advice? Do you know if they followed your paper when we were creating the vaccines?
Probably not, at least not as far as the Oxford vaccine is concerned. They got active the same time as we did. I did not know them and they did not know me. So I'm pretty sure they couldn't have read our paper. And as we know, of course, the Oxford vaccine is less effective against, it is effective, luckily, against the Lombardy type. It is effective against the British Kent variant. It is effective against the Brazilian type. It is less effective, apparently, against the South African type. Yeah, so these are changes which have to be, where the vaccine has to be modified and improved. So yes, so no, we did not know of each other, but this will be important in ongoing amendments of the vaccines.
So yeah, that would be my last question. When we talk now about the future, pandemics, epidemics, by using your method and tracing the virus, how it's going to affect the spread? Can we lower the spread of it? Can we be able to make the vaccines faster and more effective? You know, what are your thoughts?
I think the method, well, as I say, for vaccines is always relevant. We now have, fortunately, several working vaccines, but we also have variants coming up which are beginning to cause more trouble. So vaccines, on the one hand, is one application. The other one is this tracing. When I published originally, the first arrivals of the virus in the Western world had only just started. So there was still the opportunity, like in New Zealand, to shut down these infection routes by closing the flights like Italy did, and then making sure it doesn't come in. And if something does come in, you can quickly trace that person and quarantine the source. We are now in a situation where it's much too late. Nobody listened. I wrote to the government and they ignored it. So we're now in a situation where it's too late. But I hope we will soon be in a situation where the vaccines and the lockdowns will bring down the infections to very low levels again, so that we again are like a year ago, where there's perhaps one occasion there and another infection over here and one over there, and where you individually trace that and put a lot of effort into clamping down. So I hope we will soon be in that situation again, where this application of the phylogenetic analysis will become relevant again.
Thank you so much.
So Auriga, maybe you can tell a few words about your project in treatment, what you created in treating COVID-19.
Yes, thank you, Vili. I'm gonna be quick because the story is very similar. Because in March, when we got, we started to hear that COVID is now in Italy, I had the knowledge and experience by using the helmet-based ventilation, non-invasive ventilation, that it's very popular in Italy. And here in University of Chicago in 2010, actually 2014, I was writing my master's degree work about this type of interface for the non-invasive ventilation in the ICU settings. And I did the same thing. I got my family resources and created a website where I put all the information about this type of interface, how to use it, protocols, also the FDA regulations, all the manufacturers. Because what happened in the US, a lot of physicians had no idea that this type of interface is actually available in the US and it's FDA approved. So, I was trying to spread the awareness, also consulting manufacturers. One of them was NASA, the spaceship company. And now in the US, we have actually 2 more helmets that are FDA approved and are used on the patients to prevent the intubation. Just want to say I had the same obstacles with the government, with all the big facilities, with some decision makers in the hospitals where they looked at this with some skepticism. I don't know, they didn't want it to believe that it works. And especially if it was not study done in US, it's not enough data. So I went through all of that, all of it, all of this pushbacks. But as of today, the website is actually used globally. I talk with the same New Zealand because we are afraid that we will have a spike soon. People are becoming very skeptical about the virus. They don't think it exists. And people just starting to, try to live the normal life. So be afraid that we will be having a virus there very soon. And the same with Brazil, Argentina, India. Actually, we made the helmet in India. So I helped the manufacturer to create the helmet that is really good and pretty cheap to make. So that's my story.
Okay. Very interesting. It sounds familiar.
Yeah.
Maybe I can ask one question. Maybe it's not scientifically correct, but still, looking into your research and what we have heard today, would it be correct to say that it still doesn't known from where the virus comes, but most probably it is not Wuhan?
Well, I mean, where it comes from, it's really a 3-part question. The epidemic, the beginning that I showed, I would speculate China, perhaps southern China, where the bats are, possibly. Where the epidemic certainly comes from is Lombardy, and possibly with this Munich stepping stone. The question where it originally came from into humans, that is interesting that we have a 50-year gap where we don't know very much. In these 50 years, it could have been in either human or an animal. Now you would say, well, hang on, if the coronavirus has been in humans for 50 years, surely we would have noticed it. But if you think back of the AIDS epidemic in the 1980s, I'm sorry, you're probably not quite old enough, but anyway, when I was a teenager, then it was in the 1980s a new disease. But then when they started looking at older and older hospital biopsies, they found that people already in 1950 had HIV. And so for several decades, it was unnoticed. And it is conceivable the same has happened with this coronavirus, that the virus in a more harmless form has been locally in humans before it started becoming nasty. I think, however, the most likely explanation is that we simply haven't found the right bat yet. So perhaps we will find a bat with a coronavirus that is not 96% similar, but 99% similar. And that would mean it was a much more recent transmission. But perhaps this bat is dead and no longer exists, and therefore we will never find it. But the spread of the disease, I think, is possibly southern China, and the jump from the species, possibly we will never know because the bat doesn't exist anymore.
Yeah, thank you. That's quite impressive.
Mm-hmm.
So, do we have more questions?
If no one has it, I have.
Yeah, please. Sorry.
So, have you noticed that that virus is actually getting weaker too?
Weaker. It's getting worse. Our current coronavirus.
I know, but
yeah. Yes.
But maybe some strains are already getting weaker, or is it only getting stronger? Like any good news from a mutation that is happening there?
I think on the whole, it is getting more infectious. When we published, I already saw there's something funny going on with this B type. It is creating these long branches, which does not look like neutral evolution, like, steady evolution. It looks as if some of these B types are getting advantages and spreading faster and changing faster. And when I said that is a possibility, a theoretical possibility, I got a lot of criticism. Now nobody's criticizing me because it has happened. I think we have to look at two things. One way the virus can increase its efficiency, and seems to have increased its efficiency, is by shortening the time, the interval from patient to patient. And so instead of taking 5 days to jump from one patient to the next, it's taking on average perhaps 4.5 days. It doesn't sound much, but exponentially, it very quickly has a huge effect. And the other thing is viral load. If you cough and sneeze a large amount of virus, 10% more, and you have a 10% better chance of infecting someone because you have 10% more virus coughing out of your lungs, then also exponentially that will quickly make a difference, and that virus type will become dominant. And this is, I think, what we see with the Lombardy type. This is what we see with this British Kent variant. And this virus is able to do that because the spread is at an asymptomatic stage. For the first week or so, you are not seriously ill normally, but you are infectious. With other viruses, you are only infectious when you are already quite ill and show symptoms. And that also limits your spread. If you're ill, people keep some distance from you. And the virus, therefore, has a certain self-interest to keep the patient alive, so that that person reaches the stage of spreading. But this virus here does not need that. It does not need to keep the patient alive because it spreads before you get the symptoms. So this virus...
So Ebola is a stupid virus in this case.
Yes. And also the other coronaviruses were stupid because the first SARS virus had a mortality rate of 9% in 2002. That means you very quickly got symptoms and many people died. Then you have another coronavirus, the MERS virus, which comes in the Middle East from camel to human. And that has a 30% mortality rate. Yes, so that means the only way humans can spread it actually is always again and again from camels. But a spread from human to human is not very likely because you are in many cases, you are dead before you can spread it, if the mortality is 30%. But this virus has a relatively low mortality of, let's say, 0.5% or 1% or so. And therefore, the virus does not have to pay too much attention about causing serious illness to the patient. And it does that by increasing the viral load. That's my impression.
Thank you. Do you know any other virus in the history that had a similar, similar effect, similar strategy?
Well, similar strategy. I mean, Well, it's difficult to go back in history and know exactly, you know, what caused the bubonic plague, for example. You know, these are questions that scientists are still asking, even though you have very good candidates. But perhaps the most often cited example is the so-called Spanish flu, you know, where similar to today, interestingly, the second wave was worse than the first wave. And you can kind of see that the virus is perhaps calibrating itself to become more efficient. And perhaps that's what happened 100 years ago with the Spanish flu. The big difference, of course, is back then, the Spanish flu killed young people, whereas the coronavirus today is killing old people. And the reason for that seems to be that the older generation 100 years ago had experienced a similar virus 20 years earlier, and therefore had developed some natural immunity. But the young people who were born afterwards had no immunity, and they were killed off quite quickly by the Spanish flu. Yep. And today we see the opposite. But I say that as a reminder to any young people who will listen to this, that although they might now think, oh, this is a problem for older people, I don't care. These viruses mutate and they can quickly find solutions to get at you too. So it's a good idea to be on your guard.
Yeah, exactly. Thank you so much.
So
I have a question, if I may. Yeah, it's indeed, Dr. Forster, it was an amazing and capturing presentation, and I'm sure that there will be even more questions after some time, you know, because it was a lot of information. And so when you were answering to the Vilnius question about the governments, about their skeptical attitude. So basically, what that's what we see, and even without being a virologist or scientists, just active citizen. It's really easy to observe strange things and strange decisions and a lot of politics and so on. So that's like one of the why we had the second wave, you know, during elections. So it started just so.
May I ask you actually,
yes,
you who are asking the question, but also your audience. Well, A, are you happy with the handling of the pandemic in your country? And B, if you're not happy, is it because of the policies or is it because of the people? I mean, you know, a politician can make rules, but if everybody breaks them, then it doesn't work. Or everybody wants it to work, but the politicians make stupid rules. So may I ask, what do you feel is the situation in your country?
I would say that the truth is somewhere in between, actually. And we had an opportunity to spend 2 months, January and February, in Tenerife. And now we're back in Lithuania. So it's a great opportunity to compare, actually, the attitude of the people, for example, in Tenerife, and the attitude of the people here. And I was following the news in the Spanish news in Tenerife as well, and also in Lithuania. So there's also difference there. So I mean, I'm not personally happy, nor with the government, nor with the people, but I would like to go too deep to the reasons, because as a country, Lithuania is a pretty young country. So I mean, we had periods in history where we had to break rules to survive. So, and we still have a little bit this in our blood. So sometimes it helps, and sometimes it goes against us, I must say. So yeah, it's a long discussion, but yeah, both. It's also politics and also the people. I see I see young people struggling in the long lines with -3 degrees Celsius just to get into New Yorker store to do some shopping. And even though there's nowhere to go because all the restaurants are closed. So, you know, some strange things, and it feels a little bit like blockade times that we had like 30 years ago. So then we're afraid to lose this last chance to do some shopping of the stuff. So yeah. So and if I may get back to my question, then.
Yeah.
So and what would be your personal advice? But personal, of course, as a scientist. So. Since politicians are too political, and some people around us also doesn't make us too happy with their behavior. So what can we do in our households, in our lives to be more safe, and what can be our input, personal input, to stop spread of the virus and keep safe our families?
That's a very big question. I mean, I think, two of us have already tried to make a difference. You know, we just heard from your colleague in America, and me and my family have tried it using our skills. I think everybody has their own set of skills where you can do something. What I found, for example, extremely helpful were the reporters, the journalists. When we published early, there was very little research available. Some of it was in China, but in the West, nothing much was happening, because you need several months before you can set up research teams. And we got there without that obstacle. And these journalists were so helpful. They were briefing me on what was going on in different countries. And anybody, I think, can contribute to research in such a way without being a specialist. I'll tell you one thing. I was in Tenerife not long ago, and one thing I wanted to follow up was the official quarantine, where I have my doubts, because one type escaped to Italy without that being reported anywhere. I only found that out through journalists, not through scientific publications. And there is, what I haven't told you about yet, there's another escapee from quarantine. And that person, from the same cluster in Munich, carrying this, what is now the worldwide type, went all the way to opposite to where Vilja is. If she looks over the sea, she will see an island there. And that is where the person ended up, in La Gomera. And he flew for 5 hours with 200 people on the plane. Then he took a bus to the ferry. On the ferry, he went over with another 70 people around him before getting to La Gomera. So he was in touch with over 300 people, and apparently didn't infect a single person. And all this I have found out with the help of normal people like you and me. And this now gives us interesting information about the infectiousness of this early variant. How can this be the world's most infectious type, worldwide spread, but it infected nobody on the trip from Germany to La Gomera with all these people around? And is the answer, perhaps we are looking at the wrong set of mutations? Perhaps it's the more recent mutations in Italy which made it infectious, and not the other 3 mutations. Perhaps we're looking at the wrong mutation for infectiousness. And that we can only ask these questions if we understand these cases. And this is where I found journalism was extremely helpful. They pointed me to these cases. And that is something anybody can do to help researchers. You know something that could be useful, you write to a researcher. If he doesn't answer, you write to the next one. If none of the 5 or 6 you write to, perhaps your information was not that important. Yes. On the other hand, you might find someone who can make good use of that information. And that is what we all can do, I think.
Okay, then one more question, because when you were discussing with Aurika, and Aurika had a question about those underground factories, and now listening to this example about the German person flying through like a half of a globe almost, and not infecting anyone. Maybe, what do you think? Maybe it's certain habits or certain lifestyles could also affect the spread of this virus, and maybe like, and also other viruses. Some habits that we get from our homes, from our families, from the traditions, the way we eat, the hygienic habits, and so on.
It would be extremely interesting to find that out, but that means you have to interview that person. That person needs to be willing to speak to you. And the reality is, these people are ashamed. And the companies behind them are ashamed. And the governance behind them are ashamed. And they will do everything to prevent you from getting at that information. That's the situation.
Okay, thank you very much.
So, okay, thank you very much. So I think we took already a lot of Dr. Forster's time.
My pleasure.
Maybe for you was that discussion also interesting and useful?
Yes, it was.
So thank you very much for your big insights and for very interesting discussion and presentation. So we wish you good health and good success in your future work, in your researches.
Thank you.
So, Mr. President.
And on behalf of the club, I want to really thank you. That was, I think, one of the most interesting and one of the best our meetings this year. So we got really very interesting information from very first hands. So thanks for that. And as Vilija said, good luck in your further researches. And here in Lithuania, although we are almost facing the third wave, but let's stay still optimistic. And let's think that weather is getting better, it's getting warmer, so that might be the way the virus will a little bit calm down, and we will be able to get at least to some sort of normal life. So let's stay optimistic, let's be happy, and let's get not too crazy in this current time.
Okay.
And thank you, Dr. Forster, very much.
My pleasure.
Thank you.